evening, depending where you are. Thank you so much, Cyrus, for what you said. My name is Camille Granvil. With me, it's my colleague Ulrike, so we'll be focused on the regulatory consideration. Next slide, please. So we want to make sure we thank all the key contributors of these particular sessions. Next slide, please. So the agenda, so I will be focused on the comparison of the regulatory requirement across different regions and providing some of the key differences across those different regions. The second part of the presentation will be done by Ulrike, focusing on the clinical pharmacology consideration, look at some of those differences, and also in terms of the interpretation across different geographies. Next slide, please. So here, this is the landscape when we look at worldwide different health authority, North America, FDA. If you look at Europe, Asia, China, Japan, Latin America, Australia. Overall, we can say until last year, we didn't have a dedicated guidance on mass balance. On January 2024, China released guidance primarily dedicated on mass balance. FDA finalized the guidance on July 2024. So we would like to thank both health authorities to provide now. We in the industry, we have a guidance to help us when we design those studies and to make sure they met health authority requirements, whether or not we are in North America or Asia or where we are. Next slide, please. So the first questions for us is really why we do those studies, where those data fit in the drug development cycle. So here, this is a figure that has been published by Dr. Griot in 2016 from FDA. Unfortunately, he passed away last year, so we would like to thank him for his contributions in the field of clinical pharmacology. So when we look at the traditional drug development, phase 1, phase 2, phase 3, so the question is where the mass balance fit in the overall cycle. So we can say between end of the phase 1, phase 2, so we should start putting the strategy together and to perform the mass balance. However, if you are in oncology setting or there's a probability that your drug may go to an accelerated approval, that may be different. So that's why there's no one size fits all. So everything is dependent on the strategy of the asset you work on. On the right, this is just the layout. Where is the data will be fit in the US drug label. So that's been under the section 12. Those data they are providing to support the label. And below, this is just an example where we provide the percentage of the dose excreted in the urine and also in the feces. Next slide, please. Next, please. Alex? Not Alex, Jason?
I've flipped it on my end.
Okay, all right, thank you. Yes, so what we summarize here is really what we call a critical deliverables in terms of the recovery when we look at the total radioactivity in urine and feces, and also what's the percentage of the circulating metabolites. So I'm not going through all of them, but what is the key deliverable message here? When we look at across those different either document or either a dedicated guidance that has been released by FDA and as well China, so at least 90% of the dose should be the total recovery. When we look at the urine and feces, more than 80% of the recovery radioactivity should be identified when, however, metabolites is greater than 10%, the total exposure, that should be characterized. So this is really the key deliverable message when we look at all those different documents, whether or not that has been provided by those different agencies. Next, please. So here, this is a summary of when timing of the study, the design, those label position, the regulatory landscape. So we summarize everything across those different health authority, FDA, what information providing, an EMA document, Japan and China. So overall, we can say that the consensus is get the data before you start a phase 3, because those information will inform DDI, will inform which route of elimination, whether or not it's hepatic, renal. So that will help you to focus on the type of study you need in order to support your registration. So in some case, an alternative may be because you cannot do those kinds of study. So that means according to the FDA, and this is the same thing we saw in the China guidance as well, animal studies and metabolic profiling, any information that has been collected can be used to support your study. So in terms of study design, now it's really clear that 6 evaluable subject is required according to the final guidance. But in some case, it's maybe not possible. So that means you have to provide the rationale to the agency and have a conversation with them. So single dose, that's really recommended. In some cases where the time dependent, it may not be possible. So we saw the same kind of language and across those different regions in terms of those recommendations. So the dose, the final dose, the intent dose, in case the PK is linear, so an additional dose may be used to support your mass balance study. Next slide, please. So in terms of the route administration, sample collection, the analytical technique, the reporting, so we summarize, we look at all those different critical key guidance and identify what are the key information you should know based on those different health authority. For example, if you do a registration, if you have to do a global registration, do you have the data to support your registration, whether on US, Europe, China, Japan, or other countries? So the route of administration should be the route you use to support your registration. And also what is recommended is, in some cases, that can be combined with an absolute bioavailability. So there's no one size fits all. So if there's, that's something you have to look at if you need this particular study. But in some instance, the data may help you, for example, if you do a DDI, if you do a PBPK analysis, so those information may be important for your input that, for example, if you do a DDI based on the strong inhibitor, so you may use the data in addition to those information to look at moderate inhibitors or moderate inducers. So there is some benefit, but again, there's no one size fits all. Already talking about the recovery, more than 90%, and also the sample collection should be at least 90% and less than 1% in 2 consecutive days. The parent and the metabolite should be characterized. And as well, what I elucidated before, more than 10% that should be characterized. So that's really the key message when we look at across those different health authority, the type of bioanalysis that should be done both for the radiolabeled and non-radiolabeled, and the reporting that's mean all the critical key data that's mean plasma, whole blood, the profile of the metabolite, whether or not you do a pooling strategy, and how those information will be used to support your knowledge of your compound. In addition to that, where this information, what information that will be required to support your label at the end of the day. Next slide, please. So what is the key deliverable message when we look at across those different regions? I will repeat again. As today, we have 2 dedicated mass balance guidance. That means FDA and China. And other countries, they are embedded in different documents, but what we can say based on the knowledge we have so far is pretty much across those different regions seems pretty similar. There's some in terms of the expectation, that's mean the full quantitation of the metabolic pathway, the clearance, get a full understanding of what percentage of your compound is excreted into urine and feces. And those information, they are pretty much across those different regions. So in terms of what we look at some data extrapolation, for example, that has been discussed in US FDA or steady state and other those that has been in China and EMA. The study timing, what we said, it seems pretty similar across those different regions in terms of the expectation, with some difference in terms of the recommendation. That means study conduct, analysis, and reporting. So that's really the key deliverable message. And across those different regions, the expectation seems to be the same. So Ulrike, and please take it away for the next part of the presentation that will be focusing on the clinical pharmacology consideration and some difference across the different regions. So Ulrike, please take it away. Yeah.
Thank you, Camille, for the introduction. The next slide, please. Here we provided a table, and the table was filled or populated by reviewing the health authority regulation guidance of the US FDA, the Chinese FDA, the EMA, and Japan. And when we were successful and found a certain parameter, then we made a green mark. You can see here that especially between the US and China, the comparison is very similar. So actually all the parameters which we included here in the study design considerations are the same. A little bit less successful, we were finding certain parameters in the EU guidance, but there you should consider that this is not a specific guidance for human hADME. This is actually a DDI guidance. And in Japan, we found actually only one topic which was covered there, and this was the one about the radiolabeled stability. So in principle, I agree here with this finding. The radiolabeled stability is a very important part of the study. So you should consider this when you design the study. And in certain situations, you may even have the requirement that you should use 2 positions in tracking the hADME properties of a drug. For the other parameters like the route of administration, the dose formulation, using healthy volunteers versus patients, the number of subjects, the mass balance, and the PK samples, the wording was very similar. So I think we are here in an ideal situation that we do not really have to decide what to do. We really can, in principle, take, for instance, the US guidance and also compare this with the EU guidance and the Chinese FDA guidance. And then we are very well equipped with a proposal, with a guidance how to do the study. I think Camille has already mentioned most of these aspects. Maybe I pick one which was not so much covered, the dose formulation, which is of interest. So we should have here a fit for purpose formulation. We should fulfill the objectives of the mass balance study. I think that was covered in all 3 large guidances which we reviewed. On the next slide, please. You can see a summary of the bioanalytical considerations in these guidances. In Japan, there was not anything else mentioned than that what we have discussed before. But here in the US and Chinese guidance, we see also a large similarity of the individual topics which we compared. It was the quantitative analysis of total radioactivity, parent drug and metabolites of interest, concentrations and amount analysis, PK evaluations, the metabolite profiling, metabolite identification, and the metabolite structure elucidation and data interpretation. In the EU guidance being a bit less specific on the quantitative analysis of total radioactivity apparent drug and metabolites. Here we got now the information or the wish from the guidance that we should analyze the samples of plasma, urine, and feces separately for each subject and not pool across subjects. This is a very important information. And we will hear further thoughts on pooling of samples in the next presentation. On the next slide, please, we've summarized the reporting considerations. Reporting is, of course, a very important aspect of the human hADME study, because that is what goes at the end in the NDA submission. So it should reflect all the findings, and preferably it should already answer a question which may come later on. I think what is here important that you reflect the variability of the individual subjects. So that is here part 1, plasma and whole blood concentration profiles for total radioactivity, parent drug, and metabolites of interest. Also, a very good descriptive statistics should be provided. We should provide quantitative information on radioactivity, and we should estimate the absorption. So this is the same in all 3 guidances from the US, China, and the European Union. And in addition, we should, of course, present the cumulative percentage of the administered dose recovery. This is mentioned in the US and China, but I'm sure this is also the expectation of the European health regulatory boards. The biotransformation scheme and the elimination pathways is, of course, a key outcome of the study and should be presented, I think, in a very clear and thoughtful manner. And on the next slide, please. We have now provided a summary similar to that what you have seen from Camille before. So the study design on conduct of the study can be summarized across the health authority guidances, that we have a general requirement to use a stable radioactive label. In case needed, a second label might be of interest. We should enroll at least 6 healthy volunteers, and we should actually not only enroll them, they should be evaluable at the end. So if you have some drops out, you may consider to include additional subjects, or even from the beginning enroll more subjects. And we should also have a mass balance of more than 90% of the radioactivity in urine and feces. And this is very important that we also design the study in a way that we are able to achieve this. And even the health authorities mentioned, in case we see that there's a long half-life, then we should be able to prolong the study so that we have additional data which allow us to estimate the final mass balance as we are able to. The bioanalytical considerations are also here summarized. I think Camille mentioned already several of them. I would maybe once more highlight that we should really identify more than 80% of the radioactivity in excreta as with structure elucidated metabolites. This is quite demanding. I think there also we have to spend some time, but at the end we have to reduce from that the major elimination pathway, which are very important for the further use of the study in DDI studies. And at the end, the reporting, as I mentioned, very important, because that is the major reference for the study at the end in this NDA submission. The next slide, please. So these are the takeaways to remember from this part of our discussion from our work group. So overall, the mass balance study deliverables, which are summarized in these guidances, are very comparable. And we have, of course, get a guidance how to do this study. We are able to get even some information on wafers, so it's interesting to review this. And we have further criteria provided for the extrapolation on other topics. The study design on conduct is provided, 14C still being the major radiolabel which we use in our study. Still also a tritium label is always mentioned. And then the bioanalytical considerations, they are very detailed. They even ask us to think about the stability investigation of parent drug in different matrices. And at the end, the data reporting, there's also a clear guidance provided what health authorities expect in our report. Overall, I agree with Camille. We have really very good instructions now for our work. And I believe we will also have in due time an ICH guidance on this, because I think the most work is already done on the harmonization of these topics. Thank you very much. And I would hand over now to the next presenter.
So the next topic we will address today is sample pooling strategies in human hADME studies. So this topic was originally triggered by the draft FDA Human Mass Balance guidance, where the language used around sample pooling was less clear, I would say, than in the final guidance. And since we had the discussion in the technical aspects sub-team, we thought it would be a good idea also to share this broader at the outcome of the discussions and some decision trees, as you will see later in the slide deck, to share that broader. Next slide, please. So these are all the people in the technical aspects sub-team involved also in the pooling discussion, but I also have to mention that also other members from the broader IQ work group were very active, also contributing to the discussion. Next slide. So before going to our recommendations in the form of the decision trees, I want first to highlight some important general considerations. First of all, total radioactivity representing total drug-related material as well as parent drug and active metabolites always need to be analyzed separately for each subject and each time point separately. So we're only discussing here around pooling the metabolite profiling, because there pooling is often applied because of resource constraints, and a typical example is of course use of AMS, or in order to improve analytical data quality, because if you pool you can concentrate a larger sample volume, and in that way you can increase the radioactive concentration and as such also the signal to noise ratios. The number of subjects enrolled in a human hADME study is rather small, typically 6 to 8 subjects. And therefore, of course, statistical relevance of analyzing subjects individually is questionable unless there is clear variability observed. And there are other clinical studies where larger numbers of subjects are evaluated that can provide a better view on variability. And therefore, also these studies, these data can be very well used to guide the sample pooling strategy in the human mass balance studies. And as mentioned before, we propose some decision trees, as you will see on the next slides, that might be helpful in making the right choices for the pooling based on one hand data requirements and on the other hand technical feasibility. Next slide, please. So here you see the first decision tree covering pooling across subjects, where the first question to be asked is whether unusual high variability is observed in, for instance, parent PK or PK of total reactivity, dose recovery in excreta, or total mass balance. And if that high variability would be observed, then it is of course preferred to perform individual analysis. We had quite some discussion, but it's very difficult, however, to exactly define with thresholds on what is high variability and what's not. So that still needs to be discussed case by case. If that is not the case, then the next question to be asked is whether a targeted design was used with pre-selection of based on geno or phenotype. And the most typical example is, of course, the use of poor and extensive metabolizers. In that case, it is obvious that you have to analyze both groups separately, but you can still pool the samples of each group, of all the subjects in each group together. If not, then there is a question whether there are outliers observed, and I have to stress first that the term outlier in this case doesn't mean it's a statistical term, but rather is based on profiles that are inconsistent with profiles from the other subjects. For instance, if you have a clear safety signal in only one subject, for instance, and not in the others, then of course it's wise to analyze that subject separately, but you can still pool the other subjects together. And if all questions are answered no, then samples can be pooled across subjects. And although that is completely at the bottom of the decision tree, it's still the preferred and usually also the most commonly used practice. And with that, I leave the floor to Wenying, who will continue with the decision trees covering the pooling across time points on the next slide.
Thank you, Philip. So this slide shows the decision tree on time point pooling for plasma. Two decision points in this tree. The first one is based on difference between radioactive half-life and the parent drug half-life. If there is significant difference between the two, it is recommended to analyze one AUC pool plus samples from several individual time points, especially the late time point. Sometimes additional AUC pool over different time intervals may be analyzed as well. This analysis will inform late forming or slow eliminating metabolite. If answer to the first question is no, analyzing one AUC pool representing at least 90% of total radioactivity is recommended. Threshold for half-life difference is not given in this decision tree, as it should be considered on a case-by-case basis. In general, more than 5-fold difference in half-life may warrant the additional analysis if it cannot be explained by other data. The second decision point is based on has to do with the technical feasibility of analyzing AUC pool. If the AUC pool is not expected to have sufficient level of radioactivity for profiling by scintillation counting, the decision tree will point us to the alternative approach on the right. Level of radioactivity in the AUC pool can be increased by excluding late time point, and this will lead to an AUC pool that is likely representing less than 90% of the radioactivity. So we will need to generate additional data with samples at a couple individual time points, typically at Tmax and late time point. Profiling of the late time point sample by scintillation counting may also be difficult due to low radioactivity. If that's the case, LC-MS analysis can be used to analyze these samples. And of course, if MS analysis is accessible at the time of the study, it should provide sufficient sensitivity for profiling the desired samples. Next slide, please. So this slide is a decision tree on time interval pooling for excreta. Excreta samples are often pooled across time intervals based on a fixed percentage of the sample weight or volume, generating one pool for feces and one pool for urine. Typically, each pool will represent at least 90% of radioactivity in the corresponding matrix. In general, absorptions of drug can be estimated as sum of the total radioactivity excreted in urine and the total amount of metabolite excreted in feces. However, this would lead to underestimation of fraction absorbed, as unchanged parent in feces is not included in the calculation. If more accurate estimation is preferred, analysis of early and late fecal pool will allow estimating what portion of unchanged parent in the feces is result from fecal excretion of absorbed drug, thus enable more accurate estimation of absorptions. And there's another scenario in which multiple excreta pool will be useful. If the radioactivity level are too low for sample from late time intervals, including these samples in the excreta pool will lead to difficulty in profiling by scintillation counting. Creating multiple pools will allow analysis of early pool with scintillation counting and the mass spec analysis, generating mass spec response factor for metabolite, which can be used for metabolite profiling of the late pool with LC-MS analysis. Next slide, please. In summary, analysis of total radioactivity as well as concentration of parent drug and any predefined metabolite are always performed on individual samples. For metabolite profiling, it is a common practice to pool samples. However, there are situations where analyzing individual samples may be preferred or necessary. And we hope that the decision tree being discussed today will be helpful for making those choices. Thank you for your time. I now hand it over to Alex.
Okay, can you all hear me? Yeah, I hope so. So yeah, thank you, Wenying and Philip. So I will now talk about the mixed matrix method. Some of you might be asking why in a talk about human hADME are we talking about the mixed matrix method? Well, it is part of the overall metabolite evaluation that takes place around the human hADME study. But when we were originally setting up these presentations to discuss with the FDA identified this as a topic of interest to them, which is why it was included. So if you could move to the next slide, please. So as I guess you all know, the safety of drugs in development is evaluated in animal toxicology studies, where the parent drug is measured to define, among other things, the safety margins, which is of course fine, assuming that metabolites formed in these animals are the same as those formed in human. However, as anyone who works in biotransformation knows, this is not always the case. Now, there are various regulatory guidelines, collectively known as MIST, that defines what needs to be done in these situations. So for major human circulating metabolites, so those defined as exceeding 10% of the total circulating drug-related material at steady state, it is necessary to demonstrate that exposures in animals at the relevant toxicology doses are similar to or greater than those in humans, with a few exceptions, of course, based on metabolite structures. But for major metabolites which are human-specific or disproportionate, it may be the case that separate toxicology evaluation of the metabolite is needed. Next slide, please. So the definitive answer on which metabolites are in scope comes from the Human hADME study. But as you can see from this rather simplistic timeline above, many companies conduct this study later in development during phase 2 due to cost and resource considerations mainly, but also the high risk of attrition during clinical phase 1. Although this is currently changing. The main result of this, of course, is that any missed issue discovered there can cause delays with project progression. So as a result, the majority of the industry conducts exploratory metabolite profiling in first-in-human studies. This work is usually conducted on multiple ascending dose samples, or MAD samples, it's known, where the metabolite profiles can be assessed at or close to steady state. And the aim of this work is, of course, to de-risk projects before the definitive human hADME study is conducted. Now, for metabolites identified as potentially misrelevant at this stage, there are basically two options to assess coverage across species. The first is, of course, to develop a bioanalytical assay, which requires, firstly, definitive structure characterization of the metabolite, which can be tricky at that stage, as well as synthesis of a standard and internal standard. This is, of course, time and resource intensive and can even be impossible in certain cases due to challenges with synthesis for certain metabolites. There is, however, another way that we can do this, which is called the mixed matrix method, which I will describe. It's resource sparing, and I will describe it in the subsequent slides. So the mixed matrix method has been around for a while. It was the initial concept of the method was published by Gao et al. In 2010. Using they utilized a Q-trap mass spectrometry in this study, which basically was like an MRM, multiple reaction monitoring type approach, and included a statistical analysis of a set of tool compounds. So this publication involved the first proposal for the strategy for implementing this in drug development and a decision tree. The same year, Ma et al. They published as well, but using high-resolution mass spectrometry as the detection technology, which has become, I guess, the method of choice going nowadays. And of course, they also included a comparison of the method versus a validated bioanalytical assay for 2 development projects. And then in 2012, Gao et al. Followed up with their initial publication with a focus on unstable metabolites and how to address this using the method. Then later on, there was a symposium, AAPS symposium, which brought together regulatory, industrial, and academic scientists from multiple disciplines, and the method was presented and discussed there. And Takahashi et al. Published in 2017, they first coined the use of the term the mixed matrix method, and again compared the method versus the validated bioanalytical assay for 4 of their development projects. And then most recently, based on health authority feedback from a scientific advice sessions at 2 European health authorities, a team was put together to evaluate the mixed matrix method under FPS. So it was a cross-industry team, and a wide range of compounds and metabolites were tested, backed up with robust statistical analysis, which has led to a proposal of decision tree when using the method, and recommendations for best practice when performing it. This is now published as of January. The final proof is available online. Next slide, please. So how is the mixed matrix method conducted? So samples So the human samples are usually taken from the last day of dosing in the phase 1 multiple ascending dose study. The aliquots of this samples are of course first screened by LCMS to identify target listed metabolites for the later mixed matrix method. For animals, the TK samples are taken usually from the last day of dosing from the GLP toxicology study. In terms of dose selection, human, if possible, a pharmacologically relevant dose, if this is known. Sometimes it is, but if biomarkers are included in phase 1 studies, or as a worst case estimate, one can take the highest dose administered. In animals, so the no adverse event level or the the maximum tolerated dose can be used, provided if one uses the maximum tolerated dose, this is provided that any toxicities that are observed at this dose can be monitored clinically and do not pose an unacceptable risk as defined in the ICH M3R2 Q&A. So how are samples prepared? So for human, an AUC pool is prepared over the dosing interval following the Hamilton method, followed by matrix matching with pre-dose animal plasma. And for animal samples, again, an AUC pool is prepared over the dosing interval, followed by matrix matching with pre-dose human plasma. Next slide, please. So this kind of shows the experimental procedure in a bit more detail. So as you see, the first step is pooling and matrix matching the samples. Once these matrix match samples are prepared, they are then serial diluted. An internal standard is added and the samples are extracted and analyzed by LC high resolution mass spec. So, so this results in, of course, internal standard internal standard corrected LCMS peak areas, and these are simply compared across the samples to derive an exposure ratio. Next slide, please. So some comments on that last slide then. So why do we matrix match the plasma samples? So this corrects for any differences in endogenous components present in the animal versus human plasma, which could interfere potentially with the mass spectrometry response of the metabolites of interest. So why do we serially dilute the matrix match plasma samples prior to analysis? We do this to ensure that the peak integrations, the high resolution mass spec peak integrations, are performed within the linear dynamic range of the instrument. And why do we use an internal standard when preparing the samples for analysis? It's not an internal standard in the classic sense, as one would expect for bioanalysis, but it's basically used to correct for any inter-assay variability that may occur during the analytical run sequence. And in general, this, from our own experience, results in a more accurate assessment. Next slide, please. So how does the mixed matrix method compare against traditional bioanalysis? So of course, in traditional bioanalysis, a synthetic standard and internal standard must be available for the analytes of interest. And of course, with the mixed matrix method, we don't need them. The internal standard of the parent drug or a structural analog is used to correct for inter-assay variability. Of course, the traditional bioanalysis method is validated for each species without matrix matching, and concentrations of the analytes of interest are measured. In the mixed matrix method, we're not measuring concentrations of analytes at all. And if you think about the wording of the guideline, that's actually not required in most cases if it's just an assessment of coverage. The internal standard corrected LCMS peak areas of the analytes in a matrix matched animal sample are compared directly to the human sample to establish a ratio. So the final point, only animal-human exposure ratios for known or pre-selected metabolites can be determined by traditional bioanalysis. However, theoretically, using the mixed matrix method, if high-resolution mass spectrometry is used as a detection method, we could theoretically generate an exposure ratio for all metabolites detected in the human sample, provided, of course, that it is also detected in animals, and regardless of its predicted relative abundance. Next slide, please. So in terms of the results generated, so what exposure ratios are needed to demonstrate sufficient metabolite exposure coverage by preclinical species? And the ICH M3R2 Q&A contains this nice statement here. So characterization of metabolite toxicity would generally be considered adequate when animal exposure is at least 50% the exposure seen in humans. In some cases, for example, when a metabolite composes the majority of the total human exposure, it is appropriate for exposure to the metabolite in animals to exceed that in humans. So to conclude from this, then, for metabolites greater than or equal to 10%, but less than 50% of the total drug-related material exposure, an animal-human exposure ratio greater than or equal to 0.5 should be sufficient. But for metabolites comprising the majority, an animal-human exposure ratio greater than or equal to 1 should be sufficient. Next slide, please. So how accurate is the mixed matrix method versus traditional bioanalysis? It's not that easy to evaluate, actually, because, of course, we don't have any standards. But based on the available literature to date, the exposure ratios generated by the mixed matrix are very comparable to those generated using validated bioanalytical methods. On the left side, you see an excerpt from the publication by Takahashi et al., and you can see that the exposure ratios generated from both methods are extremely comparable across that set of test compounds. On the right, you see an excerpt from the most recent publication where we basically a cross-industry team compared. There was 404 measurements here, compared bioanalytical assay versus mixed matrix method for a whole set of tool compounds. And as you can see visually, they are very comparable. Statistically, there's no obvious difference between the methods. Next slide, please. So how should we implement this tool in drug development? So on the left-hand side, you see a decision tree. This is in the most recent publication. So and the acceptance thresholds here are quite conservative. As you can see, for metabolites greater than 10%, we say that the animal-human ratio should be at least 1.4 or better, or if it's the majority of human exposure, it should be greater than or equal to 1.9. If it's less than this, we advise confirmation using a bioanalytical assay. I mean, why are we conservative here? It's because of the nature of the assay. We can't validate it, right? So, so these thresholds were set based on a statistical analysis, which was done on a very large data set in the latest publication. So there's 1,544 measurements. Essentially, what it's saying here is that if you take the example on the left side, if the mixed matrix measured determined exposure ratio is greater than or equal to 1.4, there's a greater than 99% probability that the true ratio is greater than or equal to 0.5. Next slide, please. What are the advantages of the mixed matrix method? So the synthesis of metabolite standards, which can be challenging, if not impossible in some cases, due to structural complexity, is not required. As I mentioned before, the MMM doesn't measure concentrations. It's just a relative comparison between two matrix-matched AUC pooled plasma samples, and that generates a ratio. The method can be applied in early development using the leftover GLP toxicokinetic samples and multiple dose PK samples in human, thereby allowing metabolite cross-species exposure comparisons to be made at or close to steady state, which is according to guideline. Of course, one can do this maybe from extrapolations from single-dose hADME studies, but there are some complications with that. Having mixed matrix data early de-risk projects with respect to the safety testing of drug metabolites guidelines years before a human hADME study is typically conducted. If a new metabolite of interest is later, however, detected in the human hADME study that was missed in the original exploratory investigations, if one uses high-resolution mass spec to analyze the mixed matrix samples, one can go back to those raw data files later and potentially generate the necessary exposure ratio for this new metabolite. Next slide, please. Of course, there are a few recommendations for best practice. These are also found in the last publication. So this method is best conducted in phase 1 of drug development, utilizing TK samples from GLP toxicology studies, either at the NOAL or MTD, and the human PEK samples at a pharmacologically relevant dose, or the maximum dose administered if you want a worst case assessment. Samples used for the assessment should be stored in minus 80 degrees freezers to minimize stability risks. And this concept of an intrinsic stability reference, which is discussed in the publication, but also in this publication by Gao et al. In 2012, can be also used to address potential metabolite stability concerns. The analysis should be conducted using high-resolution mass spec instead of multiple reaction monitoring. This gives the most flexibility and enables reanalysis of the generated data files in the event that a metabolite not previously assessed is revealed in the human hADME study. And the recommendation is also to use an internal standard to correct for any interassay variability that may occur during the analytical run sequence. And of course, the sample should be serodiluted during the analysis to make sure that we're operating in the linear dynamic range of the mass spectrometer. Next slide, please. Of course, probably the mixed matrix method is probably not suitable in all cases. There are some situations, of course, where a conventional bioanalytical approach would be more appropriate. And the obvious one is, of course, pharmacologically active metabolites. For this, concentrations are required. A dedicated PK assessment using a bioanalytical assay will be needed to assess the overall impact on pharmacodynamic response, drug safety and efficacy, and/or the need to make dose adjustments. One other possibility is the assessment of metabolite coverage in late phase toxicology studies. So certain toxicology studies are conducted late in development, and a good example of that is the 2-year carcinogenicity study conducted in rodents. As the human hADME data is usually available at this time and the metabolites above the threshold that can potentially raise a safety concern are known, a team could consider to develop a bioanalytical assay here to measure the metabolite in the study and this to get the coverage data required. Of course, if the synthesis is not possible, one could also use the mixed matrix method in this case. And the last category probably is potentially toxic metabolites. So as we know, certain metabolites carry structural alerts that may have suspected links to adverse safety findings, such as anilines, acyl glucuronides, and several others. So if initial metabolite screening in the first-in-human clinical samples identifies potentially high exposure, the project team may prefer to develop a bioanalytical assay to monitor the metabolite throughout the drug development, preclinical, and clinical studies. Next slide, please. So just to conclude then, the mixed matrix method is now widely used across the industry. It provides a reliable and accurate exposure coverage assessment of human circulating metabolites in the animal species selected for drug safety testing. The literature shows that the exposure ratios generated are comparable to those generated by bioanalytical assay. And a cross-industry team under FPA has recently evaluated the method, has proposed a decision tree for its use, and has developed recommendations for best practice, and this publication is now approved as of January. So provided these recommendations are followed, we believe that confirmatory measurements in further studies using conventional validated BA method should not be required any longer in most cases, and that the mixed matrix data are robust enough to satisfy the available regulatory guidelines and to support registration. Next slide. And here I've provided all the literature references in case any in the audience are interested in further reading. And that's the end of my presentation. I think we move now to Q&A.
Thank you, Alex. Okay. We have a few questions that are popping up. I think the first one was for the, it looks to be, came up when Philip was talking. And so it says, talk about the preferred 90% recovery requirement, which may not be met, and ways to convince regulators that a lower recovery, 80 to 85%, for example, is acceptable.
I think that's another question for Camille in his presentation, I guess.
Mm-hmm. Oh, okay.
Yes. So indeed, if you cannot provide 90%, so you have to provide rationale why. So the agency will not really say, okay, I will not accept the data, but you have to provide the reason for that. So that's why if you have those kind of issues, the best way is really to engage as early as possible with the agency.
Okay.
Yeah. Next.
The next question is for Philip. This is about defining variability when you were describing sample pooling. Again, you mentioned about kind of how you consider it, but maybe you want to rephrase it.
Yeah, it's difficult. A very good question. I have to disappoint most of you probably because it's very difficult to answer that, and that's why we left it open and say it's more case by case that you need to decide. To give an example, if you look at percent CV, of course, you can look at percent CV of total radioactivity or percent CV of parent PK. But it can also mask, let's say, variability. If you have a low percent CV, it doesn't necessarily mean that you have low variability. If you have, for instance, two distinct groups with in each group low variability, you will end up with a low percent CV. But still, it's not a good practice to pool everything, and ideally you should then analyze these two distinct groups separately. So that's just an example that you cannot just put a percent CV threshold, for instance, and that's why we also refrained from putting any thresholds there.
Thank you.
There's a question regarding high covalent binding. I'm wondering if you can elaborate on the 80% threshold for metabolite identification. That would probably be for Camille or Ulrike again.
I can take this question, Camille. Yeah, the covalent binding is, of course, always an issue in analytical investigations. What you can do, you can try to do a very exhaustive extraction in order to really try to prove that you have a covalent binding, because already saying so is a challenge. Ideally, of course, you have also a mechanism behind it, which would explain that really a covalent binding could take place, like in case of acylglucuronides. If you do not have this, you may think about what could be done in order to get any supportive facts, any information which you can provide, which at the end would make a story out of that, what you say, and round this up and say you have done what one could, and we understand what it's there, and then an evaluation of the potential impact can be done.
Thank you.
Question for Alex on mixed matrix. How has been the regulatory feedback, FDA or others, on the use of this method?
Well, before we initiated this cross-industry team, Novartis, which is the company I work for, initiated some scientific advice sessions with 2 European health authorities, and they had some questions about it, right? Which is why we did this whole exercise and published this paper. In terms of regulatory acceptance of the data, I think there are a few examples now where people have told me that the data has been used to support NDA. So I guess the health authorities are aware of the method, and they are, I guess, gaining confidence with it with time. But one thing we want to evaluate further with this team actually is whether we can interact directly with health authorities and discuss whether any further qualification is needed.
Okay. Going back to Camille, there's one, I think, quick question. Is there a requirement to meet greater than 90% in each matrix or across all matrices?
Could you repeat the questions again?
Is there a requirement to meet greater than 90% in each matrix or across all matrices?
Yeah, it's a good question. So
For both, yeah.
Yeah.
Across both.
Across both, yeah.
The elimination should be.
Because we said urine and feces, yeah.
Exactly.
Yeah.
Okay.
That's a requirement now, Jason, so yeah.
Okay.
So this may be the final question as we're coming to the top of the hour. Philip, a question on how do you think this new proposal on pooling scheme will be adopted? How do you see the next step for it? You mentioned about publications. Is there other things you're thinking of?
Yes, indeed. So we are currently working on a publication that will hopefully come out soon with more detail, with the decision trees and more detail around the decision trees. And that's our next step, and then it's up to companies to use it or adopt it according to their standards, let's say. So yeah, give some guidance, and hopefully it's helpful to make the right decisions depending on the situation.
Okay, great. Jason, is there other questions? Is that?
There's one quick one, a little bit different. It's on the mixed matrix approach. So would the mixed matrix approach help with when justifying attempts to reduce sample volume collection in terms of 3 Rs? Does it typically need less volume of the preclinical plasma compared to individual plasma analysis?
Yeah, okay. Yeah, that's an interesting question, actually. We try to make use of the leftover toxicokinetic samples from the toxicology studies after the BA has finished. I mean, that presented some challenges, at least for us, to get that process set up, because obviously we're doing an exploratory work within a GLP toxicology study. So it took some cross-functional discussion to get that process set up and accepted. But yeah, that's the idea. The other thing that one of the companies that was involved in the FPA consortium told me that they use microsampling for this approach. So they take very small volume of plasma, 50 microliters or so from each TK time point. And then they basically dilute this in buffer and then pool the diluted plasma in buffer. If you control that well, it's no problem, and it works pretty well. Yeah, what else can I say about that? No, I think that's it. Because the toxicology doses are very high, typically, that dilution with buffer shouldn't really have much impact on metabolite detection. So there's various ways that one can approach this.
Should we go for a little bit longer, or should we conclude it?
I think we're top of the hour. So do you want to close the session, Jason?
Sure. Thank you all for everybody for attending. Greatly appreciate the audience, and thank you to all the presenters for the wonderful presentations and for the IQ for the opportunity. Have a wonderful day.
You.
Thank you.
Thank you.
Thank you.
Thank you, everyone.
Okay, bye-bye.
Bye-bye.